continuing with our last session, we will be talking about the components of embryo sacs in angiosperms. Though I had discussed with you briefly in the last class, but we could not take up all the details. So, just revising a bit of what we studied earlier, an embryo sac in angiosperms, it comprises of an egg cell, 2 synergids, a central cell and 3 antipodals. This is a typical composition of an embryo sac, which is also called as the polygonum type of embryo sac in angiosperms. Now, here you see that the egg apparatus, it comprises of an egg cell which is flanked on both the sides by 2 pear shaped synergids, and these are arranged like we tie 3 balloons together. So, you have to imagine that with threads we have tied 3 balloons together. The one which is slightly bigger and in the center is the egg cell, and on both the sides are little 2 smaller balloons which are the synergids, and then there is the large cell which is the central cell, and again there are 3 small cells at the callosal end which are the antipodals. At the micropylar end there is this egg apparatus. All angiospermous embryo sacs, they comprise of 4 groups of cells, and all these 4 groups of cells, they function in unison for fertilization, embryogenesis and even for nutrition of embryo. The minimum complement of cells that are required to effect double fertilization, which is the main purpose of this embryo sac, these cells are called as the female germ unit, or we also abbreviate it as FGU. Now, this FGU of a typical angiosperm is composed of an egg cell, 2 synergids and a central cell. Now, remember that the egg apparatus comprises of the egg cell along with the 2 synergids, and when we say that there is female germ unit, then it is egg cell, synergids and also the central cell of the embryo sac. The components of this female germ unit are involved in facilitating double fertilization. So, they are basically attracting and receiving the pollen tube, and once the pollen tube has the sperm cells, these are discharged into the synergid, and from there one sperm moves to the egg cell and the other moves to the central cell. The ultrastructure of each cell type of embryo sac we will be taking up now, starting with the egg cell, which is the most important cell of an embryo sac. Egg cell is important because egg cell is the female gamete. Now, this egg cell is sharing common walls with the synergids, like I had just mentioned, that you have to imagine that there are 3 balloons which are put together on a hook, and then the egg cell also shares its wall with the central cell. Its wall at the distal region is incomplete or absent. The wall is complete at the micropylar or the proximal end. The egg cell is polarized because the nucleus is towards the callosal end or sometimes maybe towards the center, but there is a prominent vacuole which is present near the micropyle. So, one end has the nucleus, the other end has a vacuole. Cell walls are either absent or continuous wherever the cells of egg apparatus they meet, providing direct access to the sperms at the time of fertilization. So, in the absence of cell walls, it is easier for sperm cells to move from one cell to the other cell, that is basically they can move from the synergid to the egg cell. Egg cell is richly endowed with organelles like mitochondria, plastids, endoplasmic reticulum, dictyosomes, ribosomes, so on and so forth when it is young. Now, there is an exception, that is Plumbago zelanica, where the egg cell is also endowed with filiform apparatus. Like I will be talking to you about the synergids, where we will discuss that filiform apparatus is a characteristic feature of synergids. But in Plumbago zelanica, in the absence of synergids, the filiform apparatus is present on the egg cell. Egg cell is primarily concerned with fertilization. It is the female gamete which fuses with the sperm, giving rise to zygote, which is the progenitor of the next generation. Okay. We were just talking about Plumbago zeylanica. Now, here it is the filiform apparatus which is directing the pollen tube into the egg, because this filiform apparatus is present right at the micropylar end of the egg cell. Then the other components of this egg apparatus are the two synergids. These are pear shaped structures. These two are present near the micropyle. They are closely embracing the egg cell. Now, like the egg, the wall of the synergid is also distinct at micropylar end, and it thins towards the distal end, because towards the distal end the synergids are in contact with the egg cell and also with the central cell. At the micropylar pole, the wall of the synergid is thickened to form something which is very unique to synergids. It is called the filiform apparatus, and there is a lot of diversity which is exhibited by this filiform apparatus in the synergids. They can be different in shape and also in size. So, filiform apparatus is basically a mass of finger-like projections, and they have a high concentration of secretory organelles which are present in the vicinity. This filiform apparatus behaves like a spongy wall. It behaves like transfer cells and is implicated in the transport of solutes across the sporophytic gametophytic junction through the plasma membrane. Okay, so it is basically transporting nutrients from the adjoining cells and passing on to the embryo sac. The wall ingrowths of filiform apparatus, they increase the surface area of plasma membrane, facilitating transport across the membrane. Synergid cell again is polarized, like we just discussed in egg cell. Here too the synergid is polarized with a large prominent nucleus and most of the cytoplasm being present at the micropylar end, and the callosal region of the synergid is vacuolated. Now, this is a reverse polarity which we observed in egg. Okay. The cytoplasm in synergid is also dense, and there are abundant number of organelles which are present. Now, this dense cytoplasm with an abundance of organelles is indicative of a very high physiological activity of synergids. One of the two synergids shows signs of degeneration much before the entry of the pollen tube into the embryo sac. Now, this synergid which shows signs of degeneration is called as the degenerating synergid, and this degenerating synergid is the seat for discharge of the contents of the pollen tube. So, the pollen tube enters into this synergid and then it liberates its contents. The other synergid, it does not show any signs of degeneration and it persists till the end of fertilization, and then it can degenerate after that. It is called as the persistent synergid. Now, there are certain very prominent differences between the synergid and the egg cell. So, a synergid is basically there are two synergids and there is only one egg cell which is present. Synergid is just one cell of the embryo sac, whereas egg is the female gamete. Okay. We were just talking about the reverse polarity which exists between the synergids and the egg cells. As discussed, in egg cell the vacuole is present at the micropylar end, whereas in the synergid the vacuole is present at the callosal region. Okay. Synergid nuclei are normally small, whereas the nucleus of the egg is large and it also has a prominent nucleolus which is present in that. Now, here you can see that this is a transmission electron micrograph of the egg apparatus. You can see the egg cell which is flanked on both sides by synergids. So, one synergid is very much visible and down below you can also see the central cell. The reverse polarity which we were just talking about is very much seen in this picture. Another transmission electron micrograph where we see two enlarged synergids, and the darkly staining structure is the filiform apparatus, and also the common wall between the two synergids are evident and they are shown by the arrows. In the second picture we see three antipodals which are labeled as A1, A2 and A3. The antipodals and the central cell are also connected with plasmodesmata. Here in this case, these connections are shown by the arrowheads, which are the connections present in their common cell walls. Synergid is very important and it has a very key role to perform in an embryo sac, because this is the cell which directs the pollen tubes towards the micropyle by secreting some chemotropically active substances which basically guide the pollen tube into the synergid. Degenerating synergid, like I just mentioned, it is the seat for pollen tube discharge. Filiform apparatus helps the synergid in absorption and transportation of materials in the embryo sac from the nucellus. That is why it is also called as the transfer cell. Nutrients which are received in the synergids, they move from the synergid towards the callosal end of the embryo sac. Then comes the largest cell of the embryo sac, which is called as the central cell. This central cell occupies the maximum space in an embryo sac, and it has two large polar nuclei originating at the two ends of a synocytic megagametophyte, but later on these move to the center of the cell. The two polar nuclei can either fuse completely as seen in cotton capsula etc., or they only partially fuse with each other, that is they remain in close vicinity of each other in maize, wheat and rice before fertilization. The central cell is surrounded by cell wall and a plasma membrane. Plasmodesmata occur in common walls between the central cell and all other cells of the embryo sac. The cell wall of the central cell is mostly continuous, unlike the walls in the synergids and the egg cells. Wall ingrowths, like in filiform apparatus, they can sometimes develop even in central cell as seen in soybean. The central cell cytoplasm has extensive endoplasmic reticulum and is rich in other organelles like mitochondria, dictyosomes, microbodies, chloroplasts, lipid reserves, etc. The polar nuclei are huge with enormous nucleoli indicating high degree of metabolic activity correlating with the function of absorption, metabolism and transport of nutrients from the adjacent integuments to the egg cell. The central cell stores sufficient food reserves that are later made available during fertilization and also at the time of development of embryo and endosperm. Then the other or the last type of cells which are present in an embryo sac, these are the antipodals. Okay. So, this is a picture of light micrograph where two balloon shaped structures can be seen. These are the antipodals, and this particular picture is taken in aconitum and it has 30 giant nuclei which are present. Antipodals are otherwise the smallest cells which are present in embryo sac. They are located in the callosal end of the ovule. Most organelles like the vacuoles usually are not seen in antipodals, and also antipodals have no established function and can undergo programmed cell death during the maturation of embryo sac. These are mostly ephemeral or short lived, but in some species like Caltha palustris they can be persistent, and wherever they do not die at an early stage, wherever this death does not occur, they are involved in providing nutrition to the embryo sac. They can function as transfer cells to transport the nutrient from the surrounding sporophytic cells into the embryo sac. The antipodals are known to store large quantities of starch, lipids and proteins, which are later utilized by the developing embryo and endosperm. Now, you can recall that a typical embryo sac in angiosperms, it comprises of 8 nuclei and there are 7 cells. So, it is a 7-celled 8 nucleate structure wherein we have egg apparatus which comprises of 3 cells present at the micropylar end. It comprises of an egg flanked on 2 sides by the 2 synergids. Then there is a large central cell which has 2 nuclei which are present, and then at the callosal end we have 3 small antipodal cells. Though there is a lot of variation which exists in the structure and components of embryo sacs, which we will be talking about shortly now. Another important thing about the embryo sacs is that embryo sac can also be hostorial in function. You know what a hostorium is, basically a projection and outgrowth which can penetrate other tissues to absorb nutrients. So, the embryo sac hostoria can develop from the megaspore initially, or they can develop from synergids, they can develop from antipodals, or even from the endosperm. So, the main function of the embryo sac hostoria is to penetrate other tissues so that they can take up the maximum nutrients to other tissues and pass on these nutrients to the developing embryo or even to the endosperm. The endosperm, the embryo sac hostoria perform nutritive functions for the embryo sac. The initiating cell or tissue, shape, size, course of development, time of development of hostoria is variable, and it varies in different species in different families. Very prominent embryo sac hostoria are seen in Rubiaceae and Santillaceae. Then come the synergid hostoria. Quinquamelium caelens, which is a member of family Santillaceae, shows the most extensive synergid hostoria. These synergid hostoria, they basically grow out of the micropyle into the styler tissue. They can be up to 1200 microns in length and can reach up to 1/3 the length of the style. The tips of both the synergids extend up to the base of the ovary at the 4 nucleate stage of the embryo sac. So, these long hostoria which are present in the styler tissue, they try to take up or uptake maximum nutrients from that particular area. So, you can see on the left hand side there is a picture showing the hostoria, though there are antipodal hostoria also in Quinquamelium caelens. We will be talking about that shortly. On the right hand side there is Cotodaria celona, where also there are hostoria which arise from the filiform apparatus which is present on the synergids. Embryo sac often grows beyond the nucellus into the carpel and it becomes hostorial in nature. I was just mentioning about the mature embryo sac of Quinquamelium caelens. It not only exhibits synergid hostoria but also shows the presence of antipodal hostoria in the same embryo sac. As is evident from this picture, you can see that there are 2 types of hostoria arising both from the synergids as well as from the antipodals to maximize the absorption of nutrients from the surrounding tissues. Then there are some unique ovules and certain families which have unique embryological features. Podostymaceae is one such family of angiosperms which is a very unique embryological family. Why? Because there are certain features. Now, it is very important that you understand these features which make Podostymaceae a unique embryological family. The foremost thing is that it has a 4-celled, 4-nucleate condition of embryo sac. We were talking about a typical embryo sac which is 7-celled and 8-nucleate. Here the embryo sac is 4-celled and 4-nucleate. There is occurrence of single fertilization, only syngamy. Now, this happens because the embryo sac is only four nucleate, it is 4 cells. 3 cells are the egg cell apparatus. So, 3 nuclei are present in 2 synergids and 1 egg cell. 1 nucleus is in the central cell, that is 1 polar is present. So, there is only single fertilization which is happening, that is fertilization between the male gamete and the female gamete. Now, if there is no double fertilization here, there is no formation of endosperm, though family Podostymaceae lacks the formation of endosperm. There is presence of nucellar plasmodium, which is also called as the pseudo embryo sac, which is a characteristic feature of family Podostymaceae. Pollen grains, they occur in dioids, that is they are in pairs in Podostymoideae, which is a subfamily of Podostymaceae. There is presence of suspensor haustoria which is there, and also there is lack of plumule and radicle in a mature embryo, because there is no demarcation between a stem and a root in the thallus which is present in Podostymaceae. So, basically Podostymaceae, though is an angiospermous family, it looks like a bryophyte, and it appears that it is like a bryophyte with moss capsules, that is the fruits which are present. Then there are some families where there is occurrence of naked embryo sacs, though in majority of angiosperms the embryo sac is located inside the ovule. In a few species the embryo sac is protruding out from the micropyle of the ovule. Now, when the embryo sac is protruding out from the micropyle, these are termed as the naked embryo sacs. Naked embryo sacs are seen in Galium, Utricularia, Vendalia and in Taurinia. Naked embryo sac in Taurinia, it protrudes through the micropyle, placing the synergids, egg cell and part of central cell outside the ovule, but within the ovule locule and adjacent to the placenta. The surface of the placenta in the ovary of Taurinia is covered with a mucilaginous material. So, the micropylar portion of the naked embryo sac is immersed in this mucilage. Pollen tubes grow through this mucilage on the surface of the placenta and they enter the naked embryo sac. Another important family which is worth a mention here is family Lorenthaceae. Normal ovules are absent. The ovarian cavity is occupied by a central mound or a column. It is called as the mamelon. This mamelon may be homologous with an exile placenta. The sporogenous tissue is located either in the mamelon lobes or at the base of the ovarian cavity. The resulting embryo sacs, they elongate, they move out of ovarian cavity and rise up in the stylar canal to various levels, so that fertilization occurs in the style, sometimes even close to the base of the stigma. The length of the embryo sac in Lorenthaceae, it can vary from 16 millimeter to 48 millimeter. At the 4 nucleate stage, the tip of the embryo sac, that is with the egg apparatus, it extends into the style and stigma and then it curves backwards and it becomes hooked. So, that is why Lorenthaceae is very important, and whenever we talk about a composite embryo sacs, we have to talk that Lorenthaceae is very typical. It has a very different ovarian cavity. Mammalian is a characteristic feature of Lorenthaceae. So, these families are very different. Okay. Then micropylar portion of Niscia floriblanda embryo sac shows a branched cecum. There are numerous finger-like projections which are seen in embryo sac of Exocarpus strictus, which grow in the middle of embryo sac and then they hang over the lower part of the embryo sac like a skirt. Thank you very much. We will continue with the next in the next session for the different types of embryo sacs which are available.